And it's great that it's you, Ed. And it's also a pleasure because it's the wheat lecture that I'm allowed to host, so I really appreciate that. And I've known Ed the first time I was thinking when the first time as a grad student I saw you was in 97, 96 or 97. I think it was in Spain, in Granada. Which was a crazy meeting, by the way, that Granada meeting. If that's your first meeting as a grad student, you get hooked on science. And so it's a real pleasure to introduce Ed to all of you here. Many of you probably know him already. Just a quick introduction for you, his CV. So he was undergrad and grad work he did at Delft University of Technology, if I remember. And so he did what we always consider being the worst thing, staying for the PhD at the same place where you do your undergraduate work, and was still successful.
Well, I did all the PhD work with Ray Freeman in Oxford.
I know, I did say. But there's a little twist to that. No, you were short time in Colorado as a postdoc, and since 83 or something like that, you are at the NIH, so 35 years. And Ed has worked on everything that is important for biomolecular NMR spectroscopy. Sorry to keep it that short, but that's really true. So when I started, every paper that I looked at the beginning was always, well, look first at Ed Bax's paper. And so that was, so I learned a lot from Ed Bax, how to apply NMR, how to do NMR. And so I hope we carry forward that tradition of using actually biomolecular NMR in a meaningful way for studying biomolecular NMR. So Ed has won a plethora of awards. He is a member of the National Academy of Science and all important other science academies. He's probably, for many of you, well known as one of the editors for PNAS that really helps to promote basic science in PNAS, which I always feel it's really important, and I appreciate that very much. And I will stop here because the most important thing is that we can listen to the science that he's doing, because I'm always interested in what he's doing. So thanks a lot for coming.
Thanks very much, Wolfgang, for this very kind introduction, and it's a pleasure to be back here in Tucson. I actually owe you guys, or at least the department, because one of the people in my group that I owe a lot to is actually educated here at the University of Arizona. Mike Barfield, of course, and Walker have a long history in magnetic resonance here in Tucson, and so I'm just one of the youngsters following in those footsteps. So the work I wanted to talk about today is a little bit unusual type of NMR, and I realize that for many people in chemistry, and especially biochemistry, NMR is this kind of scary technology, and it has a lot of physics behind it, and Hamiltonians, and I even get scared to mention the word in front of you. So I'm going to try to do an NMR light, and just in the middle part, hopefully make a little bit more science into it, but giving you some feeling about some of the new work that we're doing. And I shortened the title of my presentation a little bit, sort of like half as a joke. NMR does feel a little bit under pressure these days with the cryo-EM becoming the new kid on the block and being able to do all this fantastic stuff. Nevertheless, I hope to show you there's lots of new angles, new twists, new ideas in NMR spectroscopy that I'm very confident are going to keep the field thriving for many, many years to come. The project I'll be talking about today is only about a year and a half old, and we only just started publishing on it. And it's a technological challenge that started about 5-6 years ago when I had a postdoc that wanted to do NMR spectroscopy under static hydrostatic pressure, and that's a well-established field. I'll come back to that in a second. What we wanted to do was rapidly change the pressure in an NMR sample cell to see what happens. You can actually, with pressure, you can unfold the protein, and you can drop the pressure down to 1 bar, and you can try to follow by NMR how does the protein actually fold itself up. And that's a whole field in biophysics in its own right. And many people, I know of at least 6-7 groups, Switzerland, Germans, have tried to build an apparatus that can rapidly switch the pressure inside an NMR sample tube. And we're talking kilobars of pressure here. And there's one paper published in JACS about 6-7 years ago where they actually managed to get up to about 800 bar in pressure, and the switching times were way too long. They were like 30 milliseconds, 50 milliseconds. And I was talking to my colleague, Phil Anfenrood. He's a time-resolved crystallographer, and anything that takes less than picoseconds is sort of slow on his time scale. So I said, hey, I'd like to switch in a couple of milliseconds the pressure in my sample tube. And he says, well, I know nothing about that, but if you want to modify that equipment that you got in your lab, forget about it. That's not going to work. Because that's why I came and bothered him in the first place. I knew this guy is a genius at engineering. But he said, you know what, I can design and build something for you. And I said, well, good luck. If the Swiss and the Germans can't do it, you're not going to be able to do it. But he took that as a challenge. So Phil, within a matter of months, was able to actually build a rig that you'll see in a second that we're going to be using for the work I'm going to be talking about. As a bright French postdoc, a grad student that just got his PhD, like I mentioned, Jinfa Ying, who was trained here, a new postdoc, Joseph Courtney, and Ashley Barnes, who just got involved in using this for studying the formation of fibrils and oligomers of Alzheimer's beta peptide, A-beta peptide. That has been very extensively studied, and we got some nice new NMR data at the end of this, so that's sort of like still a work in progress, where you can see where this is going and what we can do with this. Like I mentioned, we'd like to study protein folding with this, and the idea is that under hydrostatic pressure we can unfold the protein, and there's many theories of protein folding. How does a protein fold when it goes from the total random coil state after it comes off the ribosome? How does it fold itself up? And there's those energy landscapes, you know, and is it rugged or is it smooth? Are there false minima on the way down? Is there a molten globule state that lives long before it finally ends up in the native low energy state? A lot of theory, and frequently shown as such a one-dimensional diagram. You start out with a lot of entropy and you end up with very little motion, or a lot less motion, but a lot more favorable enthalpy. Now this is a very serious simplification because it looks like there's only one pathway down, or perhaps if you come from the other side, obviously you can extend this, you can draw it as a two-dimensional funnel, and now you can sort of think of this like a skier. You can go down one way or you can go down a different way, and obviously this is really an n-dimensional landscape where n is twice the number of amino acids in your protein effectively, if you're sort of talking about rotatable bonds being phi and psi. Now, to a more general audience, a non-chemistry audience, I often compare this when I try to talk about this to climbing a rock wall, like this lady here is trying to get across. And what we're going to try to do is effectively the analog of what this lady is doing, study the protein climbing up the wall to its happy state where it's nicely folded. And the question really is, are there many different ways of climbing up? Or is there really only one efficient path? Or would it help if there were a little ledge here that you could take a break? Would it be faster to get to the ledge, stop for a while, and then get going again? All right, so we're going to be looking at a very small protein that had been studied very extensively. There's at least 25 papers on it in the past, ubiquitin. And it turns out that at the temperature we're going to do this, there's two pathways. One is the direct pathway and the other one is the ledge pathway. They're both about 50-50 populated. They're about equally efficient, all right? You might also wonder, what fraction of the time is the protein actually trying to fold itself if you bring it down to the unfolded state? Is it running up to the wall and trying to climb up non-stop? Or is it standing here and sort of bumping with the hand against the wall and then saying, am I going to climb up here? This looks pretty high. Or is it climbing up and falling down? And it turns out that in the analogy of the rock wall, our protein tries to climb up about 80 times before it actually succeeds and makes it across to the final folded state. And I only count serious attempts, not sort of like touching the wall, but where it actually starts climbing before it falls down. And those are like 50 microsecond, 100 microsecond events that we can measure by NMR spectroscopy. So that's the kind of information we're after for one particular model system. Of course, every protein is going to be a little bit different, but the fact that we can find this for the first protein we see must mean that there must be a more general theme behind it. Like I mentioned, we started this really earlier on with a postdoc who came out of Kathy Royer's group, Julian Roche here, who had done a lot of static pressure NMR work for his PhD, trying to look at the effect of static pressure on the NMR spectrum. And you can see here that this is what would be the proton-nitrogen 2D correlation spectrum at 1 bar for HIV protease, one of our PET enzymes. You put some pressure on it, you put a lot of pressure on it, it switches to that of the unfolded state. So the protein totally denatures, becomes totally a random coil, all right? You go halfway in between, half the protein still stays folded, it still has the same characteristic resonances, and half of the protein unfolds. So it's in an equilibrium, a slow equilibrium, because if it were fast, we would see one set of resonances. So those resonances, this protein must stay folded or unfolded for at least a second or so, otherwise it wouldn't give separate resonances, all right? So there's this slow equilibrium. Now we've got the protein unfolded. Now the real question is what happens when we drop the pressure before we get our native protein back again. So this is a couple of minutes later, after we open the valve, and it takes a while. This apparatus was very slow, like I mentioned. And so we'd like to know immediately what happens to those resonances. Is part of the protein folding before another part folds? What's going on, all right? So to do that, we had to build this new hardware that I mentioned. And this is what it looks like conceptually. It's actually a very simple device, and it's really Phil Anferud's idea of doing it the way we end up doing it. This is sort of like a copy of what you see on the right, but it's more schematic. What it contains is a reservoir, which is really a stainless steel pipe that is very strong, that can hold 2,500 bar of pressure. So it's a very thick stainless steel pipe. We pump it up. There's a pump that's hanging here off the end, and you pump this thing up to 2,500 bar. Then there's a valve here, a very strong valve that can connect through stainless steel tubing to the NMR sample cell that's here at the bottom, hanging in our NMR probe. So the sample cell is a ceramic zirconia oxide cell. It's made of the same kind of material that if you've got a dental crown, an expensive variety dental crown, it will have zirconia in it. So there's very strong material that can withstand the pressure and nevertheless be penetrable to radio frequency fields. So this we can buy. This was all built. So you open the valve here after you've pumped up the reservoir, and immediately you will compress the hydraulic fluid and put the pressure in your sample cell. Now you close this valve, don't forget, because otherwise we have a problem, and we open the low pressure valve that vents, that releases this pressure and recycles the compressed oil back into a bottle that hangs here that's then used by the pump to pump up the reservoir again. So it's a very simple kind of idea. We have two little valves here, or two big valves, strong valves, that hold the pressure and can either connect to the reservoir or connect to the atmosphere, all right? By the way, this whole thing is kept under a nitrogen atmosphere. And you might wonder why. Well, any of you who have a diesel car probably know why. If you put enough pressure on something like mineral oil or mineral spirits, that is what we're using as hydraulic fluid, it will burn. Well, it will not burn in flames, but it will actually oxidize, and this fluid turns black after cycling for a while and pumping with oxygen in there, basically just because you burn your alkanes. So the whole thing is kept under a nitrogen atmosphere while doing this. That's the only chemistry that's really happening in this experiments I'm talking about. I realize I'm in the chemistry department, so I feel it's legitimate to explain this little detail. We wondered for a while why this stuff turned black, and we thought it was the O-rings or the valves or whatever. It just turns out a little bit of oxygen. So this whole rig here lifts up, it's mounted on a pneumatic post. And you can see here a postdoc, Cyril, he's going to do a sample change. The first thing he does is he empties the reservoir. If he forgets to do that, he can blow off his foot because there's a lot of pressure in there. And there's actually some missing floor tiles underneath there and a little hole in the concrete from somebody who forgot to depressurize the reservoir before doing a sample change. So here this whole thing comes up, he pulls this thing over, and you can see this is really nice, gets it down, use a bunch of wrenches, unscrew it, change samples, and the whole reverse procedure gets it going again, all right? So this is straightforward. We can do a sample change in 15 minutes. No automated sample changers, but this is good enough. This is the actual sample tube of zirconia oxide, and you can see here this little black plug. That's actually, we use some polyethylene, black polyethylene, to separate the hydraulic fluid from the water. And it lets us see that actually you're compressing water by about 10% in volume. So if you increase the pressure here, water compresses by about 10%, so this plug comes down or it goes back up. And I'll show you an example of that in a minute. The hydraulic fluid, the mineral spirits, or mineral oil, compresses even more. That compresses by about 15% or so. So when you open the valve to release the pressure here, this mineral oil shoots up. It actually goes at about the speed of sound, about 400 meters per second. All right? So there's a fair bit of friction, heat, noise, and whatever associated with it. And here what you see is the early test runs that we did. And you see the release of pressure. It shoots a couple of hundred microliters of that compressed fluid through a funnel into this reservoir to recycle it. Okay? And that's Phil, who was overseeing this, this test procedure. I caught him on camera here. He was a little bit nervous. This was one of the early incarnations. And like I said, this plug here, you can actually see that moving up and down. If the video sometimes doesn't run, let's see if I can get it to run. Let's see one more. I may have to, oops, sorry, it didn't run. Well, we give it one more try here. Here we go. So you can actually see this black plug. It's too slow. The camera is too slow to catch it, but you'll see this thing. And one of them, you can saw it go up and down. You can't see it move because the movement itself was way too fast. We're only moving it up for 40 milliseconds at a time. So we go to low pressure for 40 milliseconds and keep it at high pressure for most of the time for this particular experiment. By the way, this was probably the dumbest thing, or probably at least the second dumbest thing I've ever done in my life, is stabilize this thing with my hand. This was before I knew that this could actually explode, and it did less than a week after this. And you don't want to be around it, so these days the whole thing is behind bulletproof glass, and that's for a reason. And I'm lucky to be alive. But you learn, you know, you're enthusiastic and you do stupid stuff. Now, the fact that we can actually switch the pressure as fast as we do, is not so easy to prove, because you can put a pressure gauge on it, but you don't measure the pressure inside your NMR sample. So for that, we can actually use the NMR signal itself. And what we're doing here is we're looking actually at the actual NMR signal, the free induction decay at high pressure, and halfway through the FID, the free induction decay, we switch the pressure to low pressure, and that means that the magnetic resonance frequency will shift. So you can see how long does it take from basically being on resonance, this is gradual decay, till it starts oscillating. You can actually plot this in the transverse XY plane, so the X and the Y magnetization. For those of you who took the magnetic resonance course, you'll see it starts changing. You can count how many dwell points it is, and from that you can count that it's about a millisecond and a half. And that's going from high to low pressure. From low to high is actually a little bit faster. You can see here, you start out at high pressure, low pressure, and we switch to high pressure, and the number of dwell points is even smaller, a little bit less than a millisecond. Now, there's a problem with that. When we switch that fast, the interface for the water-oil drops down at a speed that exceeds the Reynolds number for fluids, which means that you get turbulent mixing at the water-oil interface. So basically what you got is a very expensive, elaborate salad dressing maker. And those very tiny droplets stick around forever. So, and your protein doesn't like that. It likes to stick to those hydraulic fluid bubbles, and it's a mess. So we'd like to slow it down, and making it fast after Phil had sort of done everything he could to make it fast. I said, sorry, it's too fast, you got to make it slower. And the solution we came up with is a very simple one. We basically put a restrictor, a piece of stainless steel with a very small, about 10 micron diameter little gap in there, to try to reduce the flow of hydraulic fluid and to keep it basically at a constant rate. Now, the problem with doing that is that when you machine those in the machine shop, even while it's stainless steel, there's microscopic filings. You need a microscope to see them. You can't see them by the eye. and they come out of there and they end up in your sample tube and they mess up your experiment. And we went through this for weeks, and every time we got another filing coming out of it. And I said, you know what, we got to clean this thing out and we'll just use our apparatus. And it makes for a nice little video. We charge up our reservoir to about a third of the pressure, about 1,000 bar. We remove our sample and we just clean the valve and clean this little restrictor. So we're going to blow about 1 milliliter of hydraulic fluid out of here. There's a trash can standing underneath here to capture whatever leak comes out of here, drips out of here. And then when you fire this thing, it's in slow motion, so the whole thing really only takes a fraction of a second. This is what it looks like. And you see the stuff here blowing out of there. It looks like a lot, but it's really very little. But it's, and after you've done that, it really is clean, and you don't have any problem anymore. So this is the kind of stuff we do. It's like plumbing, you know. It's like it's fun. Problems that we have. This thing is not without fault. Everything that is mechanical and that you work on long enough, you pressurize, depressurize, everything will break. And examples of that shown here. You see this is a piece of stainless steel tubing, and it sprung a leak. All right, and it's just this tubing was good enough to withstand 5,000 bar of pressure, but this repeated banging on it, even with 2.5 kilobar, in the end it's going to go. All right, the same, we have a pump. The pump, ended up getting a bigger, more powerful pump, but see what happens here. Right, this thing springs a leak. This is a massive cast iron pump head, but in the end it dies. It goes through about 300,000 strokes before this happens. Now, to do an NMR experiment, fortunately, we only need about 10 to 20,000 cycles. So we don't replace this pump more than once a month or so that this happens. But I mean, there's always something that breaks. And that's sort of, you got to think ahead of time, is it worth doing this experiment? Because I know I'm going to have to do some plumbing and some maintenance, and it comes around periodically. I hope to show you that actually there is interesting science we can do with it. The PET protein we started on is known as ubiquitin, very widely studied. And like I mentioned, there's many NMR and other papers on it. People that have tried to study this by stopped flow experiments, quench flow. And even after 25 years, there were two camps arguing that it's a two-state folder. That means it's either totally unfolded or it's totally folded. And crossing the transition state is really a singular event, just like an enzyme, right? It's either it hasn't happened or it has happened, and the transition state is very short-lived. So the same way, this would be a two-state folder, or are there intermediates? So like, is this lady taking this little break while climbing the rock wall? Turns out that's what we're going to try to study. Now, putting this ubiquitin, under pressure, it doesn't unfold. If you take native ubiquitin, you need 5.4 kilobar of pressure before it unfolds. So you might wonder, why does a protein unfold at all when you put it under pressure? The idea behind that is that the folded protein has a slightly larger volume than the unfolded protein. So if you put enough pressure on a protein, the P delta V part, remember what that thermo was good for? That P delta V is going to drive the unfolded state now to become the lower energy state compared to the folded state that has the larger volume. At 1 bar, P delta V is nothing. But at 2500 bar, that adds up to a number of kcals. Now, you can make the delta V larger by making some holes inside your protein. All right? So if we take a valine and you make an alanine out of it, you mutate it, you basically create two cavities of about 25 cubic angstroms each. If you put two valines to alanine, you basically put 100 cubic angstrom of a hole in there. This is about 6 kcals of energy if you put 2500 bars of pressure on there. And that's enough to make the unfolded state lower in energy when you're at 2500 bar. Okay? Now, with the mutations in place, the chemical shifts are still the same as for the wild type. So this is the double mutant, the wild type. You plot the chemical shifts on top, and you see they're basically unchanged. That's because the protein retains its native structure. Even while the delta G, it's actually a little bit lower, all right? So the free energy of unfolding has dropped from about 8 or 9 kcals to about 5, but it's still a perfectly stable protein. What happens after we unfold it? Well, this is what the NMR spectrum looked like at high pressure. Actually, at high pressure, it would look with sharper resonances than here. This is 10 milliseconds after we drop the pressure, and the protein is still largely unfolded. You see there's no chemical shift dispersion here, and when we look at the amide resonances, and after it has recovered and has folded itself up, this is what the amide spectrum looks like. So we got 76 backbone amides in this protein, and each one gives its own resonance, and when it's unfolded, all of this stuff jumbles together, and it looks just like a small disordered peptide, all right, with relatively sharp resonances, although right now, while the protein is trying to fold, the resonances are broader. Okay, and I'll come back to that in a minute. Now, 65 milliseconds into it, we start seeing some of those native resonances showing up, okay? And we wait longer, we see more, and we see the unfolded resonances disappear in intensity. And you can plot the kinetics of it, and you look at this and you sort of say, okay, we've got a fast time scale and we've got a slow time scale. And if you look really carefully, you see here at the beginning, it looks like there's something funny here, that it's hesitating before it really gets going, all right? Now, this turns out to be the interesting thing. This slow recovery turns out to be a bit of an artifact of the NMR. And the artifact is that NMR is a very insensitive technique. So what we did, we were working here at about 700 micromole, 0.7 millimole concentration. And when the protein is unfolded at this point in time, it likes to stick to itself because there's a lot of hydrophobic groups. So it can actually oligomerize and give rise to an off-pathway recovery. So if we drop down the protein concentration to 150 micromole and we look at that recovery, it looks perfectly good, and this slow component disappears, all right? So this slow recovery that we see at 900 micromole, you see that becomes much more prominent. That's an off-pathway oligomer formation that is, from a perspective of trying to understand the biophysics of this protein, individual protein folding, not very interesting. Now, if you want to learn more about protein aggregation or amyloid formation or whatever, then this might actually become more relevant than the native folding of the protein. So why do proteins aggregate, and why can they actually end up in this very stable states, oligomerized states? Now, we'd like to get more specific information. This is sort of like, how does the whole protein behave as a whole? There's been a long history of studying protein folding by other methods, also NMR methods, but not the kind of pressure jump that I'm talking about. So more of the type of stopped flow or quenched flow experiments. And I'll oversimplify this a little bit. Sorry for, I know this, the whole story is more interesting and more complicated than what I'm going to be telling you, but basically what you can do is you can denature a protein at low pH, for example. Then you can very quickly bring the pH to pH 7, where the protein is going to try to start folding. And then you could drop the pH again to a level, or drop the temperature to an intermediate level where the amides, the backbone amides of the protein, cannot exchange with solvent because they're hydrogen bonded. Then you can take this protein out, you can purify it, you can concentrate it, and put it back in an NMR tube, or you can put it in a mass spectrometer and ask yourselves, you switch solvents, by the way, from H2O to D2O while diluting this. You can ask which of the backbone amide protons had exchanged with deuterium, and then you know how long did it take for an amide proton or an amide group to become hydrogen bonded during the folding process. And that's been used very extensively by Walter Englander and by many others, Carol Robinson. Many people have done this very beautiful, elegant, and informative quench flow folding experiments. Now, it was a lot of work for students or postdocs that tried to do this, because you got to first build the apparatus, then you got to harvest your protein, then you got to concentrate it, and you got to make enough of it that you can see an NMR signal for it. The mass spec makes it a little bit easier, but with mass spec, you only know that this peptide fragment or that peptide fragment was mostly hydrogen bonded or not. So you lose a little bit of resolution by mass spec compared to NMR. Now, we're trying to do the same experiment, and I'm sorry, this is the only NMR explanation I'm going to try to give, but it's not that complicated, I think. this is the NMR pulse sequence where we're actually going to try to observe the signal of the backbone amide protons. And what we do in here is we unfold it with pressure, we drop the pressure, and we start the protein folding process. But, say 100 milliseconds or 200 milliseconds after we've started the folding, I invert the water magnetization. So I don't change the solvent, I don't change D2O or H2O, I just invert the water magnetization. Now, that water magnetization, if the water hydrogen exchanges with the protein hydrogen, will invert the protein hydrogen as well. All right, so if I'm going to collect a signal here, that signal may be inverted if the amide had exchanged with water. Now, whether that's going to happen or not depends whether the protein is folded at the time that I invert my water signal. All right, so if I change this delay time tau here between dropping the pressure and inverting the water, I'm going to see a signal over here that is either inverted or is not inverted, all right, depending on how much time I give it here to exchange and depending on whether the protein had folded or not. So if all the amides become protected, hydrogen bonded at the same time, I have a two-state folder. If part of my protein becomes hydrogen bonded and the other part does not become hydrogen bonded, then something else is going on, okay? Then it's more complicated. But if the protein sort of has this single transition state, it's either all folded or all unfolded, the time constant as a function of tau will be the same for every amide that I'm observing. And this is what you see here if you look at the NMR spectra. So we've done this for different tau-R delay durations, starting out at 0. And you see many of the signals here are red. That means they had exchanged with the amide, with the solvent at 0 milliseconds, during the delay following the 0 milliseconds, that is. If I wait for 200 milliseconds, most of those guys had become protected because after 200 milliseconds the protein was already mostly folded. You remember from that curve where how fast the NMR spectrum came back? And here we can see the difference between inverting the water or not inverting the water, and it's basically an exponential decay for most of them. One single time constant. You see here Q40, Q41, two different amide groups. They're pretty much all the same. T7 is the same. The only one that behaves different here is serine 65. That seems to have a different exponential time constant compared to the other guys. Differs by about a factor of 2. All right, of course we got like 60 signals here that we can measure, and if you plot those rates as a function of residue number, you see they're all about 85 milliseconds. That's weird, actually. That's very weird. You might say, why? Because when I showed you that mono-exponential recovery, the time constant, the fast time constant, was 150 milliseconds. The only guys that come back at 150 milliseconds or close to it are isoleucine 61 and serine 65. Everybody else gets hydrogen bonded faster. That's bizarre. Now we'll come back to that again in a second, all right? And this is done at a temperature of about 25 degrees centigrade. At different temperatures, this looks a little bit different. So we'll first look at low temperature, at 5 degrees centigrade. At 5 degrees centigrade, the folding of the protein is nice and slow, and we can actually do two-dimensional NMR, directly observing how the NMR signals of the unfolded state disappear, those are the red signals, or how the signals of the blue, the folded protein, appear after we drop the pressure here. Now, those of you doing NMR, you might sort of say, hey, something sounds funny here, it's a little bit fishy. How can you take a two-dimensional NMR spectrum, say 100 milliseconds after dropping the pressure here, because it takes you a couple of minutes at best to take a two-dimensional NMR spectrum. To take a two-dimensional NMR spectrum, you've got to get many of those FIDs, typically a couple of hundred, okay? So how can I take a couple of hundred signals 100 milliseconds after this single event? Well, the trick, of course, is we cycle this many times over again. We drop the pressure, we take the signal here, we wait for a while, we go back to high pressure, and then we drop the pressure over again, and we take the second free induction decay, and we take all those guys together and make one two-dimensional spectrum out of it that is effectively taken at this time tau after the pressure drop. Okay, so it's the fact that we can repeat it over and over again. Now, we can't repeat it very fast, so while we're at low pressure, we can actually take a second or a third or a fourth measurement. All right, so these guys come for free. Well, we have to wait anyway before we can recycle our experiment. And the same here, I can look at how the folded protein, at this point it's folded because now we've been at low pressure for like 5 or 10 seconds. So after we go up, we can see how the signal unfolds as a function of time, all right? The unfolding turns out to be slower. See the unfolded signal coming back, the folded signal disappearing. That's a much slower process than the folding itself, okay? So we get 2 for 1 here in this deal. And for you right now, the only important thing is that the time constant at which the folded signal appears here is the same rate as at the unfolded signal disappears. And that basically means we observe every protein. It's either unfolded or it's folded. There's nothing missing. All right? So these two add up to unity. Now, this is pretty slow, and that's why we can do it at 5 degrees centigrade. When we go to higher temperature, this gets too fast, and we don't really have enough time to do this properly, and it gets to be very labor intensive. A smarter way of doing that is something that the NMR community would call a ZZ exchange. This is just for Wolfgang. But for you, what we're doing here is we drop the pressure, and a little while later, we measure what's the nitrogen-15 frequency. We let the nitrogen-15 evolve for a while. We measure what's the frequency, and then we do nothing for a while. And then once the protein is folded, we transfer the magnetization to proton and observe what the nitrogen frequency was a couple of hundred milliseconds earlier, okay? So we have all the time in the world here. After we let the protein fold up, we can measure the proton's frequency and correlate it with the N15, what happened here. Now, if we wait for a short time here, the protein is totally unfolded, and all we measure in the indirect N15 dimension is unfolded frequencies. If we wait for, say, 25 milliseconds, we see that some of those amide protons are still mostly unfolded, but a fraction of them, the blue peaks here, have the folded frequency. So it looks like two-state again. We have unfolded or we have folded, all right? We have two types of proteins. Some were red at this point, and the other ones were blue, and the further we move this to the right, the more we end up with folded protein and the less unfolded protein. And you can see here, you can follow the kinetics here. And you see the disappearance of the unfolded state happens with a time scale of about 75 milliseconds. But if you see how fast does the folded signal start showing up, it's 150 milliseconds. It is way slower than the disappearance here. So there's something missing, right? Because where is it gone? It's got to be one or the other, or it's invisible. Now, it's not quite invisible. You can actually see some of the invisible right here. You see this kind of little blurb here that's in between these two. That's what NMR people refer to as intermediate exchange. So this is one of the residues, lysine 11, that doesn't change its chemical shift frequency very much. And you can actually see it when it's at some stable intermediate state for about, well, you're not allowed to partition this, but I'd say 75 milliseconds, because one of them has a 75 and the other one 150 millisecond time scale, all right? So this is related now to this presence of this intermediate state. And knowing, after seeing this, knowing, hey, there is something that's invisible, there should be a way of making it visible. And one trivial idea was to look just again at the proton spectrum. Proton NMR, we had looked at those amide protons, and it's a bit of a mess. You couldn't really see much. And the resonances of this intermediate state we know are going to be nasty. They're broad because it lives very short. It only has a lifetime of 75 milliseconds. You cannot get a sharp resonance for something that exists for that shorter period of time. But if you look at the methyl group region, there is, turns out, an upfield shifted methyl group of leucine 50 in the native protein. It resonates at minus 0.3 ppm. And right after you drop the pressure, that intensity is basically zero. It's very, very low. A small fraction still of the protein had not totally unfolded, so that's what we see in here. But a little while later, this guy has a brother that resonates slightly less upfield by about 0.1 of a ppm. You wait longer, you see this guy, the little guy, getting bigger as fast as the native peak, all right? You wait longer, this guy disappears. So it looks like there is a transient intermediate with a methyl group that is right up here. Now, you wouldn't be able to tell whether those are cousins or whether it's the same guy, the same methyl group, just in different environment. For that, you need two-dimensional NMR. And again, one very simple, very old two-dimensional NMR experiment is what's called the NOSI experiment. That consists of just 3 pulsars, Jenner and Ernst and Wüthrich. Wüthrich's career was basically built on this NOSI experiment. What you do is you apply a pulse here on the protons, you let it evolve for a while, put the magnetization back along the z-axis, and then normally an NOE, that's basically distance magnetization transfer to its neighbors, takes place, and you get a spectrum with cross peaks. What happens in our case here, our protein is unfolded, we give it 50 milliseconds to start folding. Now, some of the protein will have totally folded in 50 milliseconds. Some of it is still totally unfolded, but a significant fraction of it, it turns out to be almost half, has gone to that intermediate frequency. And if we look along the vertical axis, that is what frequencies were present at the beginning of this time t1, we see there is something here at the frequency of that small methyl group that has a cross peak to the folded peak. So at the end, this methyl group is folded, but here at the beginning, it was at this intermediate position. It's this transient intermediate state that's being populated here. So it shows that this is what is known as an on-pathway intermediate, and you can draw it as a little diagram here. So we've got the unfolded state. It can either go directly to the folded state, we can follow the kinetics, we can measure the intensities and fit it to just linear differential equations. And what you get is the rates. So 8 per second it goes directly, and 9 per second it goes to the intermediate, and then to the folded state. Those rates are very temperature dependent. Now it turns out at high temperature, this is the most efficient pathway. At low temperature, that's the most efficient pathway. And the fact that people had been fighting each other for 20 years was basically because they had been doing the experiments at different temperatures. All right? So it's that simple. So in the end, everybody's right, so nobody's unhappy, I guess. Although some people are still complaining. Other interesting experiments we can do, when you do a two-dimensional experiment, what we can do is, while the nitrogens evolve, we can briefly drop the pressure and then bring it back up before the protein has time to fold. And you can ask, what is the nitrogen frequency immediately after I drop the pressure? We looked at the protons before, right? And you saw that all random chemical shifts and they're broad. But what about the nitrogen 15 frequencies? Nitrogens are very sensitive to structure. So can we measure the nitrogen 15 frequencies of the unfolded protein? And you already seen that in this ZZ exchange experiment, we can actually see them. Here what you see is a superposition of an experiment where I keep the pressure high. So the nitrogen is at high pressure, the protein is at high pressure. So just a static 2.5 kilobar experiment. Or for just this nitrogen evolution period, I drop the pressure, I see what the nitrogen frequency is when I just at the 1 bar. And what you can see here is the frequency differences. That's the red is, I've got the color coding here mislabeled. That's sort of what's confusing me a little bit. So when it's red, that's at static pressure. Blue is after I drop the pressure. And you can see there's a chemical shift difference for the nitrogen along the vertical axis. Now you would see the same if you had a small unfolded peptide, okay? You take a 5 residue peptide, you would see a 0.6 ppm change. You look at the C-terminal residue, that's unstructured in both cases, and you see this small chemical shift difference. Now the more interesting thing is like a residue like serine 20 or threonine 9 that changes in chemical shift by several ppm, all right? So that is trying to fold, but it's not succeeding, all right? It's trying to climb up that wall, but it's falling off. It has an average frequency, and these are residues that are involved in that process. Now, you can sort of already tell this gets sort of complicated. Doing the assignment on this is not so straightforward. We can actually do three-dimensional NMR on those kind of systems as well. And just very, very schematically here, what you do here is you drop the pressure. You measure either the carbon, you let the carbon evolve, or the nitrogen, or the amide proton. Then you wait for the folding to complete, and then you measure again what's the nitrogen of the folded protein, the proton frequency in a regular two-dimensional experiment, and you read out in the third dimension of a 3D spectrum what happened to your carbon or nitrogen while it was trying to fold. So we can measure the chemical shift of the unfolded system for every atom in the protein. All right, and we've about 10 years ago developed methods to try to determine structures for those proteins just using chemical shift information. Now what you see here is that here we're just looking at the correlation between the nitrogen in the unfolded state and the nitrogen in the folded state. And there are large chemical shift differences here. Okay, and they sort of correlate, but the fact that there is a correlation means glycine is always upfield and alanine is always more downfield. So part of that is residue type. That's just the covalent chemistry. There's nothing interesting. A more detailed comparison of this is sort of shown here in cartoon or in this bar graph. And what we're trying to do here, and I'm just going to focus here on, on the C-alpha chemical shifts, because those are most closely correlated with structure. C-alpha shifts downfield in the alpha helix, it shifts upfield in a beta sheet structure. And the orange bars here show you how much does it deviate from random coil, okay? So if I see a positive value here, that basically means it's transiently helical. It's unfolded, it still shows disorder characteristic, but transiently helical. Down here we see a downfield chemical shift. The orange bars are slightly downfield, meaning it likes to be extended. And how does this compare with the natively folded protein? Those are the blue dots. But the blue dots would be way off scale, so I've scaled them down by a factor of 0.2. Okay? Now if you compare the unfolded state of the protein, you see over here the blue correlates with the red. Okay? And that's what you see here. A correlation graph that happens for all those atom types, proton, nitrogen, C-alpha, carbonyl. We see a correlation with a slope of about 0.2, meaning that 20% of the time this thing behaves like it does in the native protein. These guys here, the other 50 residues, they're transiently helical, whereas they're beta sheet structure while the protein is folded. Okay? So this protein likes to be helical, even while in the end it needs to be beta sheet. So it's sort of resisting effectively here, because it's doing something that it really shouldn't be doing if it wants to fold up. But this formation of this N-terminal beta hairpin actually had been observed before, that you can see transient hydrogen bonds across those two beta strands very transiently by a former postdoc of mine, Stefan Grzesiak. And it had been speculated to be involved, and as always, Stefan was correct. So there's very transient, short lifetime. This is only about 10 microseconds we're talking here, okay? So it forms it for 10% of the time or 20% of the time. And these are very rapid events on a 10, 20 microsecond time scale. Now, if you want to learn more about the time scale on which those things happen, we have to do relaxation. NMR can measure motions, the rate, the time scales that motions happen, and you do that basically by measuring the line widths or the relaxation times of the nitrogen. That's a standard way of doing that. There's formalisms for interpreting that. And again, just for the NMR spectroscopist, we can do this. We can see very different motions for different residues when we measure the relaxation after dropping the pressure. We raise the pressure in this case to look back at the unfolded protein to find out what happened while it was trying to fold. You can do this by changing the strength of your spin lock field, and you can probe the time scale at which events were taking place. And you can correlate that, and it turns out that when you use a weak or a strong spin lock field, those events are totally correlated. And from that slope, we can extract how long did the event take. And it turns out in this case it's about 80 microseconds. And from the chemical shift difference, you can also measure what fraction of the time is it populated. And that turns out to be about 5%. All right? So 95% of the time, the lady is just looking at this wall and tapping on it and thinking, which way am I going to go up? And 5% of the time, it's actually climbing up and giving rise to this kind of relaxation. But not succeeding, because it takes 150 milliseconds, remember. So those events, 5% of the time, corresponding to 80 or 100 microseconds, you multiply it out, it turns out that it takes about 100 attempts before one finally succeeds and you jump to the folded state. So that's the kind of cool stuff we can get. There's more things we can do, but I realize I'm running somewhat behind. I want to get to the amyloid stuff. We can find out what is the structure of this transient intermediate protein. That is, while the lady's standing on the ledge. That's not published yet, but we know now pretty much for sure what's happening is that the hydrogen bonding of this beta sheet is incorrect. So one of the strands is not correctly hydrogen bonded, but it's in register. Just that there's two residues here in this loop region, isoleucine 61 and serine 65, are bungling in solution. That's why it took those guys longer to get hydrogen bonded. Everybody else is making a hydrogen bond, and when it switches from this intermediate to the folded state, there's 12 hydrogen bonds that need to get broken and be remade. But it does that without unfolding the whole protein, all right? So no wonder that it has a long lifetime of 75 milliseconds, that on a molecular timescale is infinity, of course, because it's in a very deep well. It's a misfolded, or you could say slightly erroneously folded state, because one of the strands is not totally happy and needs to jump in register, okay? So that turns out to be the reason why this protein can do what it does. Now, more interesting things that we're trying to do, or equally interesting, I should say, we're trying to do these days is try to apply this at higher fields. This was all tied to this rig was tied to a 600 megahertz instrument, and we have an 800, we have a 900 megahertz, and we'd like to do it there too. And for that purpose, Phil designed this mobile pressure device. He calls it Icarus, and those of you familiar with the Greek mythology know that Icarus is the son of Daedalus, and Daedalus was the original commercial high-pressure instrument that we started with. All right, so now you may also remember what happened to Icarus. It flew too close to the sun, the feathers melted, and I hope that's not going to be the end of our device. So here you see the two of us wheeling this over across the sidewalk, a big hazard here, trying to get this to the new, its new location. And this is the instrument that we're now using to study many of the more challenging systems. And one of those is related to amyloid formation. And I just stole this from a very nice review in Science about 13 years ago, that sort of points out, and this is like a cartoon of the disease, that we got those A-beta peptides in the brain that get released. They, in the end up in amyloid plaques, all right? And it's long been, first was believed that these are the toxic species. They accumulate, you find them at very high amounts in patients that die of Alzheimer's disease. But since then, there's been pretty compelling evidence that it's actually the oligomeric state that would be the more toxic species to the cell that might disrupt the membrane, and the precise mechanism is still a matter of debate. There might probably be multiple mechanisms by which those oligomers are toxic. But it's been very difficult to characterize them, especially by NMR. Many people have tried. By solid-state NMR, my colleagues Rob Tico, Bob Griffin at the MIT, and many others have gotten beautiful structures for those fibril states. And it turns out they form this parallel beta sheet, infinitely long ribbons, striated ribbons they're called. The oligomers, again, very nebulous, not a lot of data on it. Now, it turns out you take those fibrils, you put them under pressure, they dissolve right up. We get our peptide back again. So now the experiment is obvious that we want to do. We want to take our fibrils, or we take the peptide, you put it under pressure, you drop the pressure. First thing that's going to happen is going to form oligomers. And then we raise the pressure again, and we can observe the signal. So we can see what happened during the oligomeric state. That's the idea, okay? So if we can do this fast enough, we form oligomers within, say, a fraction of a second, and then we melt them. We can probe what happened while they were oligomers. So that's the experiment we try to do. Turns out to be a bit of a challenge. What you see here is 3 two-dimensional NMR spectra, one taken 100 milliseconds, another one 9 seconds later, and the third one 36 seconds later. And we're looking just at a very small region of the NMR spectrum. We've seen the glycine residues, a couple of serines here at the upfield region. And I've offset them, so this difference in frequency is artificial, okay? But you see that they're getting weaker, right? that means they're forming oligomers. Once they're an oligomer, they're no longer visible. They're too large. They tumble too slowly. All right? But, well, we're off by a little bit here. We've got a time scale of maybe 3-5 seconds. All right? We want to get 100 milliseconds faster than the relaxation time of our nitrogens. And then the question is, can we unfold those oligomers? And yes, we can unfold them. If we take the first spectrum 100 milliseconds after raising the pressure, the resonances are very weak. 3 seconds later, our intense signals are back. All right? So yeah, we're off by about an order of magnitude from where we want to be, but not by 6 orders of magnitude. So in principle, this should be doable. We tried to measure the kinetics, and it looks like some residues may disappear or appear faster when we go back to high pressure and disappear a little bit slower or faster, but that turned out to be not very clean. As you can see, it's not very clear cut. All right? So we had a little bit of a problem here. Another problem we're having with those A-beta peptides, in contrast to the protein, if you drop the pressure, they form fibrils. They look like beautiful fibrils. But after you repeat this about 1,000 or 5,000 times, our signal disappears. And the reason it disappears is those peptides start forming pressure-resistant fibrils. So somehow there is a morphology of fibril formation that is not sensitive to pressure, whereas those original fibrils that have been studied by everybody, they dissolve right up. But after a while, apparently there's another morphology that can take over. This is my colleague Rob Tico that kindly took those pictures for me, and this is what the fibrils look like, short stubby fibrils that are pressure resistant. All right? Now, I'd almost given up on this because we tried and all kinds of different things of making this work, making it faster, until we realized that we can actually use the fact that it is slow and that it forms an oligomer to selectively observe the oligomeric state. And here the trick is, and it's a very simple trick, we drop the pressure and our signal will disappear with the nitrogen 15 relaxation time, which is very short, unless the peptide forms an oligomer. So if it forms an oligomer, it tumbles very slowly, and if it tumbles very slowly, its nitrogen 15 relaxation will be very long. That means that if 5 seconds later we melt it, only if it formed an oligomer during this time would it still remember what its nitrogen magnetization was. So we can measure residue by residue what happened while we dropped the pressure and ignore any peptides that didn't go to the oligomeric state because they would have lost all their magnetization at this point in time. So we transfer magnetization to nitrogen, we drop the pressure, we wait for a long time, much longer than the relaxation time of a normal peptide. And then we look how much does the peptide remember that I'm going to observe here. All right, and when you do that, this is what you see. Many residues, the red contours are the residues that you observe if you would only drop the pressure for a very brief period of time, they disappear. Others retain their magnetization. So these residues that are in black here are the ones that survive. Those were residues in the peptide that became ordered. All right, and you can ask, well, how ordered were they? What was the nitrogen relaxation time? And I changed this 5 seconds to 8 seconds. I can compare intensities. I can measure T1 relaxation times. And that's what you see here. You can see how much was it retained here and what is the actual T1. And the T1 is about 5 seconds. All right, that tells us something about the size of the oligomer that we're studying. We can also measure how fast does it tumble. What is the transverse relaxation time while it's tumbling? And we can do that by putting a Hahn echo here right after the pressure drop or later on. But we're only observing oligomeric species, remember. If it doesn't form an oligomer, it's not going to be visible here. And we can see the growth of our size of our little oligomer when we move this echo pair from here to here. All right, and what you see is that the relaxation time, when it's this echo period is over here, the relaxation time is long. We're talking about 100 kilodaltons. And when we move this echo period to the right, now the oligomers have grown to about a megadalton. All right, so we can get effectively a live view of the growth of our oligomers in the NMR sample tube, and hopefully be able to sort of learn something about this initial oligomer formation at the molecular level. Well, I realize my time is up. You see the risks of working under high pressure. These were the remnants that we pulled out of a burst system. It's a rather loud and disastrous explosion when this happens. The magnet survives, the probe does not, but actually with a little bit of force you can get the probe out of your magnet, so not a problem there. This is the group that's been actively involved. You see Jinfa here, thanks to the University of Arizona. My colleague Dennis Torsja is a dear friend of mine that's been inspiring many of us. And all of this work being carried out with your taxpayer dollars in a nuclear fallout shelter near Washington, D.C. Thank you very much.
Thanks, Ed. So, a couple of things. There is a reception at Gentleman's, so there's plenty of time to ask questions. I hope many of you have time, and I would appreciate if I see many of you. We have a good discussion with different drinks in our hands. That's why it's in Gentleman's. So I want to ask the first question because the whole time I was thinking about it. So that means that this ubiquitin state measured in methanol in, what is it, 98 or something like that, that came out with that long helix. That's really the thing that you're seeing there.
It's similar or close to it. Yeah, it's called the A state of ubiquitin.
Yeah, exactly, the A state.
And that it likes that as a natural state apparently in methanol.
Crazy. Yeah. It's okay. Sorry, that was
Yeah,
when I saw that, I was like, wait.
Yep, it's very, very similar.
Yeah, it's crazy. Questions, please.
I'm actually fascinated by your oil formation, right? That salad, that really expensive salad. Could that be? This could be a really ridiculous question, but could that be what we're sort of seeing with these LPPS unstructured proteins that form this liquid phase? Could you actually be forcing part of that in these, right? And so what might be interesting is to test some of these proteins, FUS, TDP43, any of these that form these, and maybe just shake them up in there and see if you can, right? I've always been curious if you can measure this by NMR. It'd be really cool.
Yeah, actually, there are quite a few colleagues that have jumped onto this, right? Because obviously this liquid phase separation thing has become one of the hottest things in biology, and NMR is doing very nice things. Nick Fawzi at Brown, for example, is a former postdoc from our group, has been doing some of that work. We tried to find a peptide that would nicely do this and sort of see whether we could use pressure to drive this.
It would have to be an RNA-bound protein.
Yeah, exactly. And then the biology of it gets a little bit more complicated. So making a system is challenging. So we haven't gotten there. And it turned out to say, hey, I leave this up to the experts. Yeah, it's easier for me to work with ubiquitin or with A beta. But
I'd be excited to try. I mean,
likely, because there should be pressure effects on those.
Right.
So really yeah, I'd say there's potential, but it's not going to be totally trivial either.
Sure.
Man.
This intermediate, right, in ubiquitin folding that shows up at one temperature but not at another, is there an obvious rationale for why?
Yeah, I think the rationale is that it has to do with going from the intermediate state to the folded state. That the activation energy for that is such, or the barrier, that at low temperature it is fast compared to the rate that it becomes populated. So it's not like it doesn't exist, but it's a temporary dependence of those rates that causes the relative lifetime to be larger. So obviously it must exist. It doesn't get populated to an extent high enough, because going from the intermediate state to the folded state happens too fast at the low temperature relative to it becoming populated. So it doesn't accumulate to enough at any point in time. So I think the pathway likely still exists. You just can't see it.
Right, it's not necessarily that the relative stability of the intermediate is going wildly up or down.
No. It's actually, well, it is, well, the lifetime of the intermediate is relatively longer compared to the rate that it becomes populated, right? I mean, it's easy to see them if the two rates are the same, that it becomes populated and that it depletes. Or if it becomes populated and it doesn't deplete, it also would be very easy to see, because then it would be very long-lived and the other state would never happen. So it's the fact that I look at it as the two rates. So what's the faster process? It becoming populated or getting depleted? And at low temperature, the depletion is too fast. So that's why we never see enough of it.
So thanks for that, Ad. That was great. So I'm wondering how unfolded you are at the time point you picked, and if you had the pressure on for a longer time period, would you get different kinetics of refolding?
Yeah, and I hear this question more often, and the reason why I never even thought about it, because for an NMR spectroscopist it's so obvious, because once you're unfolded, you're really totally unfolded. You can tell the NMR spectrum does no longer change once you're unfolded. Although for a lot of people, really smart people, come up with this question indeed. How unfolded are you, and how do you know that the protein is really totally unfolded? If we compare it to the urea unfolded state, which chemically unfolded, the chemist normally feels is more unfolded than anything else. We look at the chemical shifts, they're smack identical. You really cannot tell a difference between a urea unfolded or a pressure unfolded, except for the intrinsic pressure effect on chemical shift, the same way you see on a linear peptide. When we put a paramagnetic tag somewhere on the C-terminus, for example, and see where does it broaden, you get the same distribution in the urea unfolded state as we do in the pressure unfolded state. When we measure the rate of translational diffusion of this cloud, this unfolded protein cloud, it measured the same radius of hydration of about 25.5 angstroms plus or minus a half. A question that's been fought over a lot in the literature is what happens if you drop the urea concentration too close to the fact where the folded state becomes populated. Do you get a contraction of your protein? Or what happens to our unfolded state when we drop the pressure to where it's almost folded? Does the unfolded protein really change its hydrodynamic radius? The answer is yes, it does, but not very much. All right, it really is a very sharp sigmoidal function, but not infinitely sharp. So you can see the radius of hydration drops by about an angstrom and a half if you're very close to the folding, unfolding midpoint. And that's because of transient hydrogen bond formation. That population of transient hydrogen bonds compacts the protein a little bit. But at high pressure, it really is very unfolded. You can do it for a week or for a millisecond. Once it's unfolded, it's totally unfolded.
Yeah, so back to the amyloid fibrils, so you were able to identify the residues that are involved in the intermediate formation. And you said that if you repeatedly do these pressure jumps, it transitions into something that then withstands pressure and doesn't basically dissociate anymore. So my question is, have you done the experiment sort of sequential times to the point where you're getting close to those fragments that basically can withstand pressure and identified additional residues that might be sort of facilitating the interaction of those pressure withstanding?
It would be a very nice experiment to do, but it's very difficult because once it doesn't come back, we're out of luck. We can't see it anymore.
Really a sort of a step function. There's no sort of
well,
transition or
for us, the peptide is visible or it's invisible. There's no peptide that we can see part of the peptide. So once it doesn't let go of the oligomeric state, it's black. We can no longer observe it. So only the guys that totally let go and are totally loose and happy, unstructured, are visible to us.
So maybe the other question would be, if you repeated the experiments, you continually see exactly those residues sort of repeatedly showing up as forming the oligomer, or if as you do the experiment, like if you could, let's say, do this as a repeated process over time, would you start seeing some changes in that or not? I mean, to maybe get some insights into how it might sort of transition into this thing that all of a sudden becomes resistant to pressure?
Yeah, I think the answer to that is not with the methods that we're using, because for us, the fact that we see some residues go into this oligomeric state doesn't mean that not the whole peptide goes into this oligomeric state, but those N-terminal 10 residues are somewhat disordered, all right? So they have a T1 that goes up to 1.5 seconds, but not to 6 seconds or 5 seconds. So the whole peptide gets ordered, but not enough to retain its magnetization at the end when I make it visible again. So if it goes to this pressure-resistant state, it never comes back.
Yeah.
I'm never going to see it anymore. I see nothing. So I would need something else.
So this might be a naive question. So if you're thinking about the pooling for ubiquitin, and there are two different pathways depending on temperature, what's the sense that if you're folding, say, on the ribosome, is it single pathways, different pathways, or the surface, like everything that folds is probably folding very close to something that's very large and dense. Would this change? Is there a way to test this in the system?
Yes and no. What happens on the ribosome is obviously, I mean, it's going to be sequential. So for large proteins, they require the ribosome, or they're going to require some chaperone to fold it up. For us, the way to test it in a very primitive way is to add, we can do this basically in a cellular environment. Right? Now, ubiquitin is not very suited for it because it likes to stick to all kinds of things. So the NMR spectrum looks pretty crappy if you put it in a cell lysate. But we've done that, and actually if you put it in cell lysate, you look at the recovery of your protein, we see less of a population of this slow recovery state. So the whole spectrum looks sort of pretty marginal, but we can go to 1 millimole, and we don't get those aggregated state that we see for the pure protein. So somehow having this whole soup in your NMR tube that makes the spectrum look lousy actually has a positive effect on the recovery of the protein. So what happens in the cellular environment effectively is more complicated than this model system that I'm showing you here. And that's, I guess, what the lesson is, that you look in a test tube, but is it really what's happening in real life? No, probably not. And adding cell lysate is not the same as looking in a live cell, but it's sort of like a very primitive poor man's mimic of what I can do.
Well, I was told that there is an exam will be held soon here, so we should move over to the other venue, which is all good. So thank you again, Ed.
All right.
Thank you.